The purpose of the photosynthesis light intensity lab was to determine the rate of photosynthesis by measuring the effects of varying light intensities on a moneywort aquatic plant. In this experiment, the independent variable was the intensity of light between the light source and the moneywort aquatic plant at 20 centimeters, 40 centimeters, and 60 centimeters. The dependent variable was the rate of photosynthesis by measuring the displacement of the volume of water of the aquatic plant in a span of 3 hours. The control variables were the volume of water, the length of the plant, and the mass of baking soda being used. The materials that were required to conduct this lab were 3 1-liter beakers, a moneywort plant, 3 small graduated cylinders, 3 small funnels, a 500 ml beaker, baking soda, a lamp, 1 meter ruler, marker, tape, a petri dish, a scapula, scissors, a storing rod, a stopwatch, tweezers, and an electronic balance. The hypothesis that was produced for this lab was that if the light intensity increased on a moneywort aquatic plant, then the displacement of the volume of water will be greater. To begin with this experiment, we first gathered all the materials that were required to execute this lab and labeled 3 1-liter beakers in regards to their distances from the lamp. In each of the 1-liter beakers, we measured out 1,000 milliliters of water by using a 100 ml graduated cylinder and poured it into each of the beakers. We later measured out 3 grams of baking soda on a petri dish using an electronic balance and added it to one of the 1-liter beakers containing water. We repeated this process 2 more times so that 3 grams of baking soda had been added to all of the 1-liter beakers containing 1,000 ml of water, and stirred the solution until the baking soda had fully dissolved. We took another separate beaker and measured out 500 milliliters of water using a graduated cylinder and mixed it with 1.5 grams of baking soda and stirred the solution. We later took a stem from the plant by measuring and cutting the length to 10 centimeters and repeated this process until we had achieved 6 stems 10 centimeters each. Using these stems, we trimmed the ends off of all 6 stems by cutting them off to an angle. We positioned the 3 separate beakers in their designated distances from the lamp, so with the first one being 20 centimeters away from the lamp, the second one 40 centimeters away, and the third one 60 centimeters away. And we made sure the lamp was off at this time. Subsequently, we placed 2 trimmed stems in each of the beakers, ensuring that the cut side is facing upwards. We placed a funnel on top of the 2 stems in each beaker. We filled up each of the mini graduated cylinders with the water and baking soda solution from the 500 ml beaker and covered the top of the cylinder with our finger and flipped it upside down, making sure that little to no liquid escapes. We repeat this process for the other 2 1-liter beakers. We turned on the lamp, ensuring that no external light source was present, and recorded quantitative and qualitative observations before and after 3 hours. The objective was to measure how much water was displaced that would later indicate how much oxygen was produced from the graduated cylinder placed on top of the funnel. We observed that before the 3-hour period began, the 3-liter beakers containing the plant were all equal in temperature at 22 degrees Celsius, and that the starting volume of water and baking soda solution were all at 1,000 milliliters. The 3-liter beakers contained an equal amount of 3 grams of baking soda, and all the liquid mixtures were transparent. All the beakers also contained an equal measurement of 10 centimeter stems, with each beaker containing 2 stems. All stems were green in color, however, each of the stems contained a different number of leaves. After 3 hours were over, we observed that for the beaker that was 20 centimeters away from the light source, the temperature increased from 22 degrees Celsius to 30 degrees Celsius, making the beaker feel warm. There were more bubbles visible in the sample, and 3 milliliters of water had been displaced. The sample that was 40 centimeters away from the light source, we observed that the beaker felt somewhat warm, and the temperature only went up from 22 degrees Celsius to 23 degrees Celsius, and there was only 1 milliliter displacement of water. The sample also showed a similar number of bubbles in the solution as the 20 centimeter sample. Lastly, the beaker that was 60 centimeters away from the light source did not show any significant change with 0 displacement of water. The temperature of the water remained constant at 22 degrees Celsius. However, there was a tiny oxygen bubble at the top of the cylinder, and there were a lot fewer bubbles in the solution. This light intensity experiment helped determine the rate of photosynthesis by measuring the displacement of water in the graduated cylinders. The addition of baking soda in the 1-liter beakers filled with room temperature water helped to ensure that the concentration of carbon dioxide was not a limiting factor, which allowed to showcase the effect of light intensity on plants. Aquatic plants such as moneywort plants would release oxygen when they undergo photosynthesis, and the rate of photosynthesis would be determined by measuring the amount of oxygen collected that had been displaced with water. Photosynthesis in plants is triggered by light, and in this experiment it indicated that as the distance between the moneywort plant and the light source increased, then the displacement of water was lower, meaning the rate of photosynthesis decreased. For instance, these following results were taken after waiting 3 hours in this lab. The observations indicated that when the beaker that contained the moneywort plant was 20 centimeters away from the lamp, the volume of water decreased by 3 milliliters. At 40 centimeters, the volume of water decreased by 1 milliliter, and at 60 centimeters, there was no indication of any displacement of water except for a tiny oxygen bubble at the top of the cylinder. These results indicated that as light intensity increased, then the rate of photosynthesis also increased as the volume of water being displaced was greater with the oxygen gas, since a higher energy of light was being lit towards the plant. Therefore, more oxygen was produced and more carbon dioxide was being used, until the carbon dioxide usage had reached its light saturation point, which caused the rate of photosynthesis to become constant due to a limited number of rubisco enzymes that were needed to allow for photosynthesis to occur. A plant contains many specialized organelles for photosynthesis called the chloroplasts. Chloroplasts contain chlorophylls which give plants their green color. Inside of the chloroplast contain thylakoids, which are in stacks called granas, and on a thylakoid membrane is where photosynthesis is initiated when the light hits these packets of pigments called photosystems. Photosystems consist of an antenna complex which contains accessory pigments such as chlorophyll B, xanthophylls, and carotenoids, which are the first pigments that absorb the high energy coming from the photons, as they have the ability to withstand that high amount of energy, whereas chlorophyll A cannot endure that high amount of energy coming from the light and can potentially denature. The light transfers between the accessory pigments in the antenna complex, and once the energy becomes lowered, then the energy is absorbed by the reaction center, which contains high concentrations of chlorophyll A. This energy is used to move 2 electrons to a high energy state, where it is later captured by an electron acceptor in the light reaction. This process is called photoexcitation, which is needed for photosynthesis to occur. Additionally, the light reaction is one of the 2 steps in photosynthesis which helps produce the products needed in the Calvin cycle, such as ATP and NADPH, that is later used to produce G3P and converted to glucose. When the 2 electrons are captured by the A0 electron acceptor, then the 2 electrons need to become replenished in the reaction center to prevent chlorophyll A from denaturing. This can be achieved through a process called photolysis, which is the hydrolysis of water using light to extract 2 electrons from water, and these electrons are later used to replenish the 2 electrons lost in chlorophyll A. Along with the 2 electrons being produced, 2 hydrogens and half a water are also produced. The hydrogens contribute to the electrochemical gradient in the light reaction, whereas oxygen can leave the chloroplast and can either be used in the mitochondria for cellular respiration, or it can leave as waste into the external environment through guard cells. Therefore, if a higher intensity of light was being lit to a plant, then photosynthesis would occur at a higher rate, which would produce more oxygen to leave the plant due to photoexcitation and the light reaction occurring at a higher rate in the chloroplast, and for photolysis to also occur at a higher rate. An experimental error that could have occurred during this lab was that there may not have been enough leaves undergoing photosynthesis, and there may have been an unequal number of leaves on each stem, which would have resulted for the displacement of water to be potentially low, and for the volume of oxygen accumulated to also be low. Each of the 3-liter beakers contained 2 stems in 10 centimeter length from the moneywort plant, which was trapped under the funnel. However, all of the stems contained different number of leaves as well as different sizes of leaves, which could have affected the rate of photosynthesis, as a varying concentration of oxygen would have left each stem depending on the size of each leaf and the different number of leaves on each stem. For instance, the beaker that was 60 centimeters away from the light source did not showcase any displacement of water, potentially due to a low number of leaves causing less oxygen bubbles to be released from the leaves resulting in a lower displacement of water. To conclude, the purpose of this photosynthesis light intensity lab was to determine the rate of photosynthesis by measuring the effects of varying light intensities on a moneywort aquatic plant. The hypothesis that was produced for this lab was that if the intensity of light increased on a moneywort aquatic plant, then the displacement of water would be greater. This hypothesis was accepted because, as per the experiment, the observations confirmed that within the 3-hour period, when the beaker containing the 2 stems of the moneywort plant was 20 centimeters away from the lamp, the volume of water decreased by 3 milliliters. At 40 centimeters, the volume of water decreased by 1 milliliter. And at 60 centimeters, there was no change in the displacement of water except for a tiny oxygen bubble that was observed at the top of the cylinder. These observations proved that as the light intensity increased, then the rate of photosynthesis also increased as the volume of water being displaced was greater with the oxygen gas. A recommendation that could be made to improve the performance of the light intensity lab would be to precisely measure and count the number of leaves on each of the stems, and for them to be an equal amount to ensure that the same number of leaves undergo photosynthesis in each of the 3-liter beakers to receive more accurate measurements of the displacement of water and the accumulation of oxygen gas.